Take it away.
Okay, that's wonderful. Okay, John, focus. Do what you did before. Let's see. Okay,
No
I go
pressure.
over here, and you can see nothing but the title slide. Is that correct?
Correct. You're good to
Yes.
go.
Okay, excellent. So yes, I have visited the Truro campus several times over the past couple of years, and I would have loved to do that again this time. Hopefully, circumstances will change a bit, and I can visit in person soon. You're quite right. Genomics, I think, is foundational across this, swath of research and industry portfolios at modern universities. And so today I'll give you a sense of, what my particular research interests are and what my lab does, but hopefully at a high enough level that people can see some room for synergies, perhaps around the technologies used and so forth. So that's what I'd like to do today. So yeah, I'm a basic, good old-fashioned biologist by training. When I was an undergrad in the late 90s, I became smitten with molecular biology and the idea that DNA sequences were able to store information and be used for answering all manner of questions, including those in the realm of evolutionary biology. This is just a cartoon of a tree of all of life, I suppose, on here. What it shows you is a couple of things. Hopefully, the density of organisms for which molecular and genomic data are available, number one. But also, some of the organisms that I'm particularly interested in and fond of are the protists near the top right. These are single-celled eukaryotic organisms, and although they're very somewhat marginalized in this particular diagram, they represent the bulk of the genetic and I would say biochemical and molecular diversity of eukaryotic life. And so that's another theme that runs through my research interests straight from graduate school to today. And I want to set the stage for thinking about some of the take-home messages with this quote. It's from a colleague of mine in the US. And this person, Chuck Delwich, wrote a really nice article almost 10 years ago, well, more than 10 years ago now, almost 20 years ago. And he referred to the genome as being like a palimpsest. And if you've ever heard that word, it's an ancient text that has been written on early paper or some sort of predecessor thereof. And oftentimes the information would be written on it in different directions. So if you stare closely at that image, you can see there's text running both vertically and horizontally multiple times over and over again. And so the idea here is that the genomes of modern day organisms are like palimpsests. So they retain information, and that information is written over on top of one another over and over again as organisms and their genomes and their molecular biologies evolve and adapt in response to their environments. And it's just a lovely visual image for how I think about genomes as storers of evolutionary information. And you'll see some of these themes coming through in what I'm going to present today. I'm not going to assume that any of you are experts on the tree of life or indeed genomics and molecular biologist, but what I am going to do is tell some sort of high-level stories about our work and the power of modern genomic technologies for addressing fundamental questions in biology. So the first part is to do with genome mosaicism. So this is the idea that the genomes of any given organism, including human beings, genomes are mosaic. So they are mixtures of genes from different evolutionary histories. Sometimes those origins are ancient. We now know that they are also ongoing, and that's a theme, again, towards the end of the talk. And they're certainly puzzling. So I'll set the stage for the middle part of the talk by just telling you a little bit about how we think about mosaic genomes and how we detect mosaicism. I'll then kind of dive into this phenomenon of lateral gene transfer, so the exchange of genes across species bounds rather. How important is that? How does it happen, and so forth. And then I'll give you a little bit at the end about some baby steps that we've been trying to take to develop systems to actually study LGT in the lab. Now, my PhD advisor was this person, Ford Doolittle, and I was in his lab between 1997 and 2001. And right around that time, he was making a splash on the international stage for his work on lateral gene transfer. And this is an image from a Scientific American piece that he wrote. You can see the title there. You can see a tree cartooned, and what you will notice is the fact that this tree is not strictly bifurcating. It is networked or web-like in structure, and this is this cartoon is really coming from the realization that strict divergence of lineages over time without any subsequent interactions is an oversimplification of the way that cells and organisms evolve. And really, this realization came from the comparative genomic era. With the more genomes we have collected over the past 20, 25, 30 years, the more interesting the story has become, but also the more confusing certain aspects of it have become. So it was an exciting time to be a graduate student at that time. And boy, things have really changed in terms of the technologies and the questions we're addressing. Now, the title of my talk today, part of it, if I remember, involved highways of gene sharing. Now, I stole that part of the title from this paper. Again, an old paper from a former undergraduate who I knew at that time, and now a colleague here at Dalhousie in computer science, Rob Biko. This is just a sort of state-of-the-art paper in 2005 analyzing 144 different prokaryotic genomes, basically everything that was available at that time. 144, more than 200,000 proteins, and they sought to identify pathways of genetic exchange. And so each of the circles in this cartoon is a bacterial group, and the lines that connect the different circles, the thickness of the lines and the numbers associated with them indicates the number of genes that were postulated to have been exchanged between the members of those circles. So the highway analogy is that there are highways, that is proper, regular modes of exchange, and then because there are highways, there are byways and much smaller avenues. So just with a relatively small sample size like this, you could make some inferences about how organisms are passing genes around and which organisms are interacting with which other organisms, and how frequently that's taking place. This has led to the phenomenon of what's referred to now as the pangenome concept. And I'm going to use our lab friend E. coli as the textbook example because it is stunning and relatively straightforward to think about. You might think of E. coli as a lab strain. Of course, the standard one that we use for molecular biology and cloning and expression and so forth, the basic strains, minor variations thereof. But of course, E. coli is all over the world, in our guts, out in the environment. And the amazing thing is that the more E. coli genomes that have been sequenced, the more we realize how different they all are from one another. Now, the cartoon on the bottom right, what it is showing you is along the bottom axis is the number of strains, E. coli strains, that have had their genomes sequenced. And as of this paper, there's about 3,000 or so. So 3,000 different E. coli genomes to compare. And then the vertical axis is the number of genes we can roughly consider that number to be. Gene clusters is a minor nuance that we won't worry too much about. Just think about it as the total number of genes. And here's what we see. Every time you sequence a new E. coli genome, you find new genes in it that have never been found in any other E. coli genome thus far. Okay? And furthermore, the more sequencing you do, occasionally you will find another E. coli genome that doesn't have any of the genes that all the other E. coli strains that have been identified has. So this gives rise to this concept of a pangenome and a core genome. So the core, if you look at the Venn diagram on the left, the core is the genes that are found in all or nearly all of the E. coli that are in your comparative data set. And then the accessory genome are the ones that are present in the periphery of the Venn diagram. Okay, so again, the amazing thing is the more you sequence, the more you find novel genes that have never been found in E. coli so far. We're at 3,000 or so E. coli genomes. It's more than that now. And the total number of genes that have been found in at least one E. coli is more than 40,000. And this is amazing because there's no individual E. coli has ever been found that has more than about 4,500 to maybe 5,000 genes. So again, it's telling us that even within bacterial genus and even species, in quotations, we see a surprising amount of genetic variation. And we know very well now that those genes are, those sort of accessory genes are the product of lateral gene transfer. So they're coming from other non-E. coli organisms, and they help those strains adapt to their environments, be it the human gut or on skin or in soil or something like that. Okay, so that's the concept of the pangenome as applied to prokaryotic organisms. Now, eukaryotes, you and I are eukaryotes. We have, our cells are more complex than prokaryotes, so we have a nucleus and more complex internal features, a cytoskeleton, endomembrane system, and so forth. The nuclear genome of eukaryotes has long been recognized as a mosaic. And the bulk of that mosaicism comes from the precursors of the mitochondria and the chloroplast, or the plastid. So you and I have mitochondria, and those mitochondria are derived from once free-living bacteria. And mitochondria have a genome, a very, very tiny genome. In the case of the human mitochondrial DNA, it's about 16,000 base pairs. Plants and algae have a chloroplast. That's what those little green balls are in that cross-section of a leaf. And they are derived from a different type of bacteria, cyanobacteria. Chloroplasts have DNA too. The genomes are generally a little larger than mitochondria, but still very, very small compared to cyanobacteria, the bacteria they come from. And we know that organelles, mitochondria and chloroplasts, are dependent on about 1,000 or more proteins, generally. Proteomics experiments confirm that, and yet their genomes contain anywhere from about a dozen genes, sometimes even less, up to about 200 at most. And what that tells us is that during the course of integration of those precursors, of the bacterial precursors of mitochondria and chloroplasts, genes moved from the mitochondrial and chloroplast precursors into the nuclear genome. So that is a phenomenon called endosymbiotic gene transfer. And that is an example of, I guess, what I would refer to as a highway, a gene exchange highway. It's has occurred to a great extent in the evolutionary history of eukaryotic cells, and still occurs regularly today, including even in organisms such as ourselves. Occasionally, pieces of mitochondrial DNA end up in the nuclear genome of humans, and sometimes they unfortunately cause disease and so forth. So that's a highway. Now, the uncertainty in the case of eukaryotes comes from where other genes in nuclear genomes come from. And there is, everything else is sort of lumped into this category of lateral gene transfer, also called horizontal transfer. And lots of uncertainty there. Is it feeding that gives rise to these genes, that is, eukaryotes engulfing other types of organisms? Do these genes come directly or indirectly via viruses? There's evidence for both of these, but by and large, it's bioinformatic evidence and much less robust. And so there is a lot of uncertainty about eukaryotic nuclear genome biology and evolution, and the extent to which pangenomes are somehow, whether the pangenome concept is meaningful for eukaryotes. Now, I mentioned endosymbiosis earlier on when I was talking about mitochondria and chloroplasts. Things get really, really complicated when we think about the history of photosynthetic eukaryotes. And this is because we can have multiple waves of endosymbiosis. So this is a case where an organism can come to reside inside another organism, and that outer organism can itself be taken up by a third organism. And in some cases, given enough time, these three different organisms can become completely cemented or meshed together, cell biologically, biochemically, and even genetically. And so we have genes being transferred by EGT at various stages, and also there is the added complication of horizontal or lateral transfer. So nuclear genomes in many lineages of eukaryotes are completely and utterly mosaic, and that's what these colorful chromosomes are meant to represent. And what that really means is that when we think about the eukaryotic tree of life, and this one is skewed a little bit differently to emphasize the photosynthetic lineages and the single-celled organisms, what we have are a lot of uncertainties. And I'll just buzz through these arrows here without boring you with the details. If this looks like a giant hairball with colors on it, that's a pretty good description of it, which is to say that we're good at identifying the colors. So we're good at identifying photosynthetic lineages and the basic type of chloroplast that they have. We're not good at inferring how exactly that organism came to possess those chloroplasts. So chloroplasts or plastids are patchy. They appear all over the eukaryotic tree, or much of it. And part of the problem is because the genomes, the quality of the genomes that we're analyzing are also patchy, that is, of poor quality. Okay, so that's a bit of background on laying the groundwork for what I'm going to tell you next, which is a little bit more about eukaryotic LGT and how it is that we're trying to better understand it as an evolutionary force. Now, here's where we're going to tell you a little bit about the technology. Something that has really changed the way that my lab and many other labs do what they do is called long-read sequencing. And so there is a company called Oxford Nanopore, and probably about 6 or 8 years ago now, they started releasing a tiny instrument called a MinION device that is, well, you can see a picture of it there next to a laptop. You know, basically could fit in your back pocket. And this machine, this instrument, is incredibly powerful. I'm going to pause this because it gets kind of annoying. Oops. Let's see if I can do that again. Essentially, the way this instrument works is the instrument has these hundreds of these biological pores through which DNA substrates are threaded, and the instrument is able to detect the signatures of the different nucleotides in DNA one by one as they pass through each of the biological pores. It's incredible that it works, but it does. And the main advantage of this is that the individual reads, the snippets of DNA sequence that we get from the instrument are much, much longer than those that come from previous types of instruments. Okay, so by long, I mean 100, 200, even close to 1,000,000 base pairs per individual read. And this gives us much more power in our ability to piece together the genomes of organisms in a much less expensive fashion. Now, they come in all different shapes and sizes now, these nanopore sequencers. Some of them are a little bit more scaled up and would be more sort of the size of a toaster. And you can get even smaller ones that you can actually hook up to your smartphone. Why you would exactly want to sequence DNA on your smartphone is not entirely clear. It's a bit of a gimmick in that regard, but this company has done their homework in the marketing business. They've sequenced DNA on the space shuttle. They contributed to sequencing viral DNAs in an Ebola outbreak and so forth. So they've demonstrated that it's a very flexible and powerful and relatively cheap and transportable way of sequencing DNA. We can buy these instruments for about $1,000, the ones that we use in my lab. So relatively inexpensive. Now, how are we using that? So really what nanopore sequencing and other forms of sequencing over the past 5 to 10 years, what's really happened is that we can start to ask much more fine-grained questions about how genomes are evolving over time. And what this shows you is just, again, a generic tree of eukaryotic life with different lineages highlighted. And some of the ones that we're particularly interested in are highlighted with these little bubbles, okay? Which is to say that because genome sequencing is so cheap and inexpensive today, in terms of cost and time, we can much more deeply sample individual lineages. Individual strains and species belonging to particular branches of the tree, in the same way that people have collected all those E. coli genomes that I was discussing earlier. And when you have that kind of fine scale resolution, it allows you to ask questions that you simply cannot ask or answer when you only have sort of one genome from this branch of the tree and another genome from that branch over there. This is much more fine-grained comparisons and therefore much more powerful in terms of getting to the question of mechanism. And that's what I'm going to tell you a little bit about today. So, and we're going to focus just on a little bit of a highlight reel on a group of single-celled algae called pelagophytes. And you can find them pretty much everywhere, in the sea primarily. The pelagophytes are part of this larger eukaryotic group called stromanopiles, and you don't need to remember any of these names, but it's actually quite a large branch on the eukaryotic tree. And for our purposes, what's interesting is that it encompasses photosynthetic organisms, like the ones I'm going to tell you about, pelagophyte, but also diatoms and giant kelp even. Then there are mixotrophic organisms, so they photosynthesize and they eat things. And then there's heterotrophs that don't have chloroplasts and make a living by other means. So we chose this system specifically because there was this high density of reference genomes relative to the pelagophytes in some of these other lineages, but not in the pelagophytes themselves. So this arrowhead is showing you where we've focused. Now, by we, I mean Shannon Sibbald and Maggie Lawton. This is a project that Shannon started probably 5 years ago now. She's probably going to defend her PhD in the next year or so. And so what I'm describing is unpublished data that we're really, really excited about. Shannon and an undergrad, Maggie, used nanopore sequencing for a variety of different strains within this lineage, 5 different strains within one genus, in fact, one species, Oreococcus anaphogeferens, and then 2 other ones that were slightly more removed. This one here, Pelagomonas and Oriumbra. And what do we do with this? Why would we want to do this? Well, again, the idea is that when you have this fine scale resolution, you can ask questions that about mechanism and frequency of gene transfer that you simply cannot ask when you have a much sparser sampling. And that's what these different arrows on this diagram are meant to represent. And the goal, the long-term goal, is to integrate the data we have on strains, species, genera, and lineages within this stromanopiles and get this synthetic picture, hopefully, of how genes have come and gone in that lineage and how frequently that process is. So just some numbers to tell you just how much of a difference this nanopore sequencing that I mentioned, how much better does that make our data? Well, we're actually resequencing genomes insofar as there was one published in 2011, which is not that long ago. It's a small genome by human standards, 53.5 megabase pairs. Using the technology available at that time, it was broken up into 5,000, more than 5,500 different discrete pieces or contigs. And that's a function of, by and large, the limitations of the technology used at that time. So it's a good picture of the types of genes, the number of genes the organisms have, and roughly the size of the genome, but not a high resolution product. We redid this exact same genome with nanopore sequencing, and the number of fragments went down from 5,700 to 142, plus completely sequenced the organelle and the mitochondrial DNA. So very, very effective. We combine the long read sequencing with more traditional short read sequencing to improve the accuracy, and we also do RNA-seq or transcriptome sequencing to tell us which genes are expressed and to help us identify the genes in the genome more effectively. I mentioned, so we have 5 different anaphogeferens genomes, which I'll tell you a little bit more about in a second. But then Maggie Lawton, again, Maggie was an undergrad, and so this tells you how fast the tech is going. Part of her undergraduate thesis was to completely sequence 2 nuclear genomes. And when I was a graduate student, I think I sequenced about 15 genes in total, probably for my entire PhD. So things have really, really changed. Now, again, to underscore the power of this nanopore tech, in this case, the one on the right-hand column here, the number of fragments, like the sizes of the individual contigs that were assembled was a total of 8. And we originally thought, okay, this can't be right. The number is just too small. But it turns out that, in fact, Maggie's sequencing in her assembly was correct. She had completely sequenced and assembled all 6 chromosomes that the organism has, 6 and only 6 in the nucleus, telomere to telomere, so completely resolved all 6 chromosomes, and sequenced also the plastid and the mitochondrial DNA, effectively with one run on an nanopore sequencer plus some Illumina sequencing. So we celebrated this result because no one in my group or any other group that I know of at the time was able to do anything quite like that. Looping back to Shannon's work on the strains of Oreococcus, she took the lead from a group in the US that had looked at different strains associated with an Oreococcus bloom in the coastal waters near New York. And long story short, these are the locations of these strains, and in particular, whether they were found to be sensitive to a particular virus that is known to infect Oreococcus. So we strategically chose these strains, 1, 2, 3, 4, 5. One of them is the original one that was sequenced in 2011, and then 4 new ones. So all of this, Shannon did basically all of this work herself, one PhD student, all the DNA extraction and culturing, all the sequencing, all the assembly, and all of the downstream analyses. And I'm going to tell you kind of what this picture actually looks like when you compare all of those data. Now this is a Venn diagram, and this should, I hope, remind you a little bit of that E. coli one that I mentioned to you, or showed to you earlier. This is how people, at least for small numbers of strains, compare and search for the pangenome. So you have 5 strains, and then the numbers in the different portions of the Venn diagram correspond to the relative frequency in the different categories. So there's 16,000 and change number of proteins or genes found in the genomes of all 5 strains. Then we have other sort of subsets of that, and then at the extreme are the ones where there is the genes are only found in the individual strains and not in any of the other 4. And when you look at those numbers, it adds up to almost 10%. So 9% of the genes in Shannon's analysis are strain specific. And I should say too, you might be wondering what I mean by strain. How different are these strains? Well, genetically, when we look at sort of reference genomes, or reference genes rather, to get an estimate of how similar they are genetically to one another, they're typically 99% or greater, even up to 99.9% identical in terms of the DNA that they actually share. So these are not divergent strains, they're recently diverged strains, and yet they still have about 9% of their genes not found in any of the other 4 strains. When Shannon looks at these so-called accessory genes, we see that they are enriched in interesting types of genes, both in terms of the functions and also where the genes come from. So many of these strain-specific genes are bacterial genes, not eukaryotic genes, as best we can say. So they appear to be coming from other organisms that Oreococcus is interacting with by lateral gene transfer. This is not unprecedented by any means. This is a recurring theme when we look at eukaryotic microbial genomes. So bacterial genes are not new, but it is very satisfying to see this correlation between the strain-specific nature of these genes and the sort of foreign source from LGT. Now, one quick point here has to do with viruses. Now, one of the things that Maggie did for her honors thesis that ended up in a publication was to look at the organella genomes. They were sequenced as well, and she identified a particular region in the enophageferens strains. I'm going to show you, it's this red arrow. And that was curious to us because it wasn't found in the genomes of the other species that she had sequenced. And when we zoomed in there and looked at that a little bit more closely, what she found were DAM-methylase genes that appear to be of viral origin. And so I'm showing you just, this is a phylogenetic tree of one particular one, a DNA cytosine methyltransferase. And what you can see is the gene in red here, highlighted red, that's the mitochondrial homolog, and its closest neighbor in the known universe of sequences is in fact in the viral genome of the virus that is known to infect Oriococcus. So this is clear evidence that that virus that is preying on these algae in nature is, at least in some cases, donating DNA to the alga itself. One final example to make this point. This is a study that's in press now, done by a postdoc of mine from Korea, Jong-in Kim. And long story short, she was looking at another lineage of algae. And you can see those little circles. These are chloroplast genomes sequenced. And we find quite a size range in the chloroplast genomes. And one of them in particular, which is highlighted up here, something called fibrocapsa, has this somewhat bloated chloroplast DNA and with particular regions that are of foreign origin. So she found evidence for these, some of the genes that are listed on the bottom left. We won't go through the details, but they're like no other genes that are found in this entire rhaphidiophyte clade of algae. And in fact, they're most closely related to, in some cases at least, plasmid DNAs that are found in diatoms. So we don't understand this by any means, but it's suggestive of somehow in nature this particular rhaphidiophyte is interacting with diatoms and perhaps picking up bits of plasmid DNA and integrating that into its plastid genome. So another example of LGT into eukaryotes. So let's just return to this highways and byways analogy. This is a synthetic cartoon that I hope will give you a sense of what I've been telling you. So again, this is a cartoon of a eukaryotic organism. The highways of gene sharing or gene exchange that we're familiar with and understand, at least to a large extent, mitochondria and chloroplasts have donated genes extensively to eukaryotic nuclear genomes. Somewhat less so are endosymbionts, so more transiently associated bacteria. There is some evidence for that kind of transfer as well. And then everything towards the bottom here is much, much more speculative. So viruses, conjugation between prokaryotes and eukaryotes, that has been documented and can actually happen. And also just DNA coming from food, so ingested prokaryotic and eukaryotic organisms, sort of along the lines of you are what you eat. Those are very much uncertain in terms of the precise mechanisms and frequency. So lots of unknowns here that still need to be discovered. And so that's the last little few slides here, and that's to tell you a little bit about how we would like to be able to get to a situation where we can actually study this phenomenon of lateral gene transfer in the lab. This work was started about probably 6 or 8 years ago. And we've had some modest successes, but many failures as well. As many of you appreciate, to do experiments in the lab requires you to have some kind of a model system that you can work with. And for various reasons, we chose an organism called Acanthamoeba castellanii. It's an amoeba, as the name suggests. It's got a relatively small genome. We can grow it on plates. We can grow it in liquid. We can transform it. And importantly, it's known to host many giant viruses in nature. So many of these new viruses with large genomes have come from this particular organism, Acanthamoeba castellanii. And one of the things we needed to do to convert this into a better model system was to improve the quality of the reference genome. A bit like Oreococcus earlier, this genome, I think, the Acanthamoeba genome was published, I think, 2013, something like that. And it was a good start based on the tech at the time, but we wanted to make it better so that we could do a better job at studying gene transfer in the lab. So long story short, this paper is under revision at Genome Research, and we've effectively produced chromosome scale genome assembly using nanopore and related technologies for the original strain and a closely related strain as well. And this is just some of Morgan's work showing some of the similarities and differences in the genes that we are finding. Like Oreococcus, we see even between closely related strains, they differ in their gene content. Now, that was only part of the story because what we really wanted to do is to develop a system whereby we could feed Acanthamoeba genetically transformed organisms and try and build a system where we could detect the movement of genetic material from the food into the nuclear genome of the organism. All of those constructs have been built and tested. As I said, we can transform the organism. You can see there in the fluorescence images. We can also select single cell isolates of the organism. That is, when you want to get to the point where you completely sequence a genome, we can take little small samples of a transformed cell culture and plate them out, just basically put them onto a plate of E. coli, and then we just let the amoebae crawl across the plate on their own, as you can see in the bottom left. So they mow their way through this lawn of E. coli, and then we can pick up the individual cells on the end, and we can drop them into little liquid cultures and then grow them up. And we know that they're clonal from that point on, and we can sequence their genomes. I have a postdoc as well who's about 8/10 of the way to producing a system for modifying the genome of Acanthamoeba using CRISPR-Cas9 technology. And the goal here is to really have a way to manipulate the gene content of the organism so that we can start to sort of dissect some of the molecular and cell biological factors that might underpin the ability of organisms like Acanthamoeba to pick up foreign DNA. So we're still in tool building phase and have had no success in actually demonstrating lateral transfer in the lab as of yet. So to conclude, tree of life or web of life, is the eukaryotic component of the tree of life more like a tree, or is it more like a web as we see in bacteria and archaea, or prokaryotes in general? And the answer is pretty much yes. This is an essay that was written by a very talented undergrad a few years back. He's now a PhD student of mine in the lab, where we summarize kind of taking stock of what's known about these phenomena as it relates to the evolution of prokaryotic and eukaryotic organisms. In the case of eukaryotes, there is, I think, very little doubt that LGT is not quantitatively, at least, the factor that it is for prokaryotes. There is a much greater trend towards verticality, sort of traditional vertical evolution in eukaryotes. Certainly, we see that in the multicellular lineages, animals, plants, and so forth. But even in the microbial realm, I think the signal is much more vertical than it is for prokaryotes. But nevertheless, there is sort of web-like components, and I think I've presented some of that evidence to you today with our studies on different algae. And you know, as comparative genomicists, we're always in the business of collecting more data. The more data we collect, in some cases, the less we actually understand. So we're at a really exciting time in this particular field to be able to collect amounts of genome sequences that would have been unimaginable 20 years ago and start to address some of these fundamental questions. So I will stop there and leave it at that. And certainly thank you for the invitation and for your time today. And I'd happily address or answer any questions. Thank you very much.
Thank you, John. I'm an economist, so I'm always amazed by genetics. It's mind-boggling to me, literally. And I turn to my colleagues all the time to help me straighten that out. Before I go off, yeah, okay. So, Fraser, let's start with you. Do you have a question?
Thanks, John. I'm wondering, with these, you talked about 9% of the genome, I think maybe of the Aerococcus. Yeah. Sorry, I work on Aerococcus and you're working on
Oriococcus, yeah.
Oriococcus, yeah. So 9% is sort of in that pan realm. Initially, I thought, wow, that's cool. That must be 9% new stuff that they use. But then I'm realizing there must be a lot of junk that gets transferred that has no utility. Is that the case?
Yeah, that's a very good question. The short answer is that we don't detect as much junk as we would expect to see. And this is one of the mysteries of this particular project. Certainly that suite of genes, that 9%, I think virtually all of those genes in those individual strains are expressed. So there's no reason to think that they are junk in the strict sense of the word. And it is also the case that, you look at the biological features of those, the properties of those genes. They can tell you some interesting things, and you can make educated guesses like, okay, that gene, it really makes sense that organism might have that particular gene to help it grow in a certain way or something. So, that's evidence supporting that those genes are functional, at least over short time scales. The junk, this is where the quality of the genomes really comes in, and why it's super nice to have fully resolved genomes. What I didn't have a chance to tell you about today is that we do find the scars of past interactions with viruses in the genomes. So there are fully integrated chunks of Oriococcus viral DNA in the nuclear genomes of some, but not all, of those Oriococcus strains. And we even find bacteriophage stretches of DNA. And bacteriophage, of course, in fact, bacteria, not Oriococcus, but if Oriococcus is eating bacteria and taking up bacterial genes, then perhaps this explains where they're getting some of this phage DNA. And those fragments of DNA very much do look like junk, insofar as they're not obviously coding for anything, but we can detect them because we have such high quality reference genomes. So we see some junk. I would have expected to see more, but I also, and we're limited by, the lens that we're looking at this problem through. We just don't know based on because we don't know the frequency, we don't know how much junk to expect to see at this kind of a scale. So I hope that answers your question.
Yes, thank you.
Mm-hmm.
Thanks, Fraser. Goddard, go ahead.
Yeah, thanks, John, for the great presentation. The point is, I'm not sure even this is relevant to the topic, but the point is, I'm curious to see that when we see common genes between even species, how likely is they have the same functionality?
Mm-hmm. The same genes in within a species or between species?
If you comment within a species, and even like that would be better.
Yeah, that's a tough question to answer, but I think it's an important one. And I would say it's common, and I think it's probably the norm, but it needn't be. I think that's the interesting thing about it. People who study genome biology and gene function and gene diversification, anyone who's dug into that knows that gene duplication is a driver of new functions. So, you've got gene A, and then that duplicates within the genome. Gene A and A prime. And then if they drift apart from one another, they can acquire new functions. So there's a lot known about how genes acquire new functions. But, gene A, as I just mentioned, that could be found in all organisms. It could be found in all members of a species. It could be found in all eukaryotes. And yet it is conceivable that the sequence divergence that we're seeing is to a certain extent underpinning new functions. Now, then it gets complicated, and we say, well, what do you mean by function? Like, how different do they have to be to count as a new function? Like a similar function, a completely different function? So there's a lot of gray zone there. It's an excellent question. It's a really tough one to answer. And really what we do with these types of experiments, we're just making estimates about functions based on sequence. So anytime I tell you, that's a damn methylase gene, or that's a this or a that, it's because its sequence looks like something that has been characterized as such in the database, and that may or may not be a good assumption to make, but oftentimes it's the only thing that you can do. It's a good question. Yeah. I see Chris has his hand up. Hi, Chris.
Hey, John. Great to see you virtually.
Yeah, good to see you.
Yeah, I hope to see you again in person soon.
Mm-hmm.
So I started teaching first-year biology this year, and I'm talking about things like endosymbiosis theory. Yeah, and I've actually mentioned you and your work with classes in that area. Giving you props with my class. And one of the things we talked about is, a little bit at a rudimentary level, obviously first year course, is horizontal gene transfer, vertical gene transfer. And I remember I'm looking at the figure here right now. I talked about this, how horizontal transfer in the textbook anyway talks about it might have been much more prominent in the past than it is now. And one of the comments you made at the end of your talk was that it's maybe not as prominent, at least as we thought, as vertical gene transfer. Has there been a difference over millennia, you think?
I think there has. It's a good question, and I think there has, and I think it varies from lineage to lineage. And another version of this talk would have unpacked some of that complexity. Let's just say, and I'm aware of the time, I don't want to blab on too long, but you mentioned chloroplasts. So let's stick to that example. So we have a plant cell, yeah, with a chloroplast inside it. And that chloroplast, like Arabidopsis, say, has, I don't know, 100 protein genes or something. So, the progenitor of that chloroplast, the cyanobacterium that gave rise to it, had 1,500 plus genes. It must have. and yet the chloroplast only has like 100 some. So that's the sense in which that's an ancient transfer, probably before plants and algae, when they were first evolving, there would have been this massive influx. So that would have been a highway, like a freeway. But over time, what we now know is that rate of transfer slows down. So in endosymbiotic gene transfer, it's probably the case that there are these periods of time where it's just raining down on the nuclear genome, and then it slows down. And lineage to lineage, there's lab experiments showing you that in tobacco, it's still extremely frequent for reasons that I don't have time to explain. You look at a field of tobacco, and like 1 in 10,000 tobacco plants has got a fresh piece of chloroplast DNA in its nucleus. And then in other lineages, you go and look, and you can't find any evidence of it. So it's very dynamic. So this is the ancient is known, the ongoing is known, but the uncertainties relate with how one switches between both of those two extremes. Yeah, good question.
Good.
Thank you.
Thank you, Chris. And Yan-Fei for the last question, because we are getting really close to 1. Thank you.
Thank you, John, for the seminar. My question will be really quick, and maybe I can even follow up with you later. So I guess my question is how we can collaborate. For example, if we are interested to using the facility, and I think it can be challenged. Like I wonder if there's a specific bioinformatics team in Halifax campus, for example, using RNA sequencing as an example. After we isolate RNA, there's still like sample preparation, for example, the library preparation. And then if there's a bioinformatics team to do the alignment to the reference genome, and then the researcher in agriculture campus, we're doing the downstream analysis, because for people don't have a bioinformatics expertise, that can be very challenging. For example, we got the raw data, now how we can do the downstream analysis?
Yeah, that's an excellent question. And Yan-Fei, I remember you were in attendance at the workshop that we ran a week ago, maybe 2 weeks ago, tomorrow. And this is something that I've been pushing for a long time at Dal, and I think we're starting to get some traction. So Chris knows this as well. I'm really excited about trying to build these bridges between different corners of the Dal community, and that includes the ag campus. So by all means, there are opportunities for making these formal connections, and I think it starts with the dialogue. What are you working on? What technologies are you using? What barriers are there for you to carry out your work? So let's follow up, and I'll come to Truro soon and hopefully meet more of you. That would be wonderful. Thank you.
Thank you.
Excellent. Well, John, this is great. I really, really we all appreciate your time here today. And I think if there's anything we can do to help to set up some bridges, that would be, let us know, because I think that would be really, really appreciated on both sides.
Yeah, wonderful. Okay, thanks everyone. I appreciate your time.
Thanks so much,
Enjoy
John.
the rest of your sunny day, and good luck with your field work.
Happy long weekend.
Yeah, you too.